Okay, in this video, let's talk about making some male gametes. So to do that, let's talk about why the testes are outside the body and the scrotum. Because when the baby boy is made, the testes start in the abdomen. They're the same organs that become the ovary in the woman, and it's not until the end of the second trimester, beginning of the third trimester of pregnancy, that they start migrating down and out down in the pelvis and make their way through the abdominal wall into their final position in the scrotum. If they were to remain inside the abdomen, no sperm would be produced because the temperature is too high in the abdomen. So what happens is the arterial blood that's traveling down towards the testes is surrounded by this network of veins. And so there is a heat transfer between the colder venous blood returning to the trunk of the body and the warmer arterial blood. There's a heat exchange going on. So by the time the arterial blood reaches the testes, it has dropped in temperature so that it will not be warm anymore. All right. So looking at the testes, as I said previously, there is a tough white outer coating that comes inward and divides it into compartments, and each compartment is called a lobule. And inside each lobule, there's going to be 1 to 3 tubules called seminiferous tubule. And each of these tubules is about 6 feet long, like fingertip to fingertip. And it's inside the seminiferous tubule that the sperm will be made. So here is a cross section of a seminiferous tubule with the lumen in the middle, and there's the next one, and there's the next one, and here is a scanning electron micrograph. And so here on the outer part is where the germ cell precursors are located, and as they start dividing and undergoing meiosis, they pull away from the outer part and go towards the lumen. And then here in yellow, these are the sperm which have formed, which are then going to travel in this tubular system and make their way into the epididymis. All right, so let's look at the cells of the testis. So this is the cross section showing multiple seminiferous tubules, and this is a cross section showing a wedge of 1 seminiferous tubule. So when you look at it, these purple cells inside this black circle are the same as these purple cells inside the black circle, and these are the germ cells in various stages, which I'll talk about on a subsequent slide. As you can see, the germ cells in this drawing are surrounded by these yellow cells, and these yellow cells are Sertoli cells, and I'll talk about them on another slide as well. I do not have a higher power picture here where I can show you where the Sertoli cells are, but they are there. You can see like there's more pinker cytoplasm in some areas, and that's to show the Sertoli cell parts. And then surrounding on the outside of each seminiferous tubule, there are these other cells right here, which are called interstitial cells. If you took this course from somebody else, they may call them Leydig cells, but I call them interstitial cells because they're between. Inter is a prefix that means between. So they're between the seminiferous tubules. And these interstitial cells are the cells that are going to be secreting the androgens, the number 1 androgen being testosterone. So these are the cells that make testosterone, whereas inside the seminiferous tubule, those are the cells that make sperm. So you can see the two are not related to each other. They're next to each other, but they are not the same cells. So let's look at their Sertoli cells first. Okay, so Sertoli cells are kind of like nurse cells. Okay, as you can see, they are surrounding all of these lighter blue cells, which are the developing germ cells, all the cells that are going to be gametes. Okay. And in fact, you can see right here and here and here, there are tight junctions there. And if you remember way back at the beginning of the course, when we talked about intercellular junctions, tight junctions means nothing can get through here. And so the biggie is these developing gametes are going to be going from diploid to haploid. And if a macrophage came along, it would be going, whoa, that's not normal. And it would want to phagocytize it and get rid of it. And so this is going to prevent those types of immune reactions from happening within the testes. Sertoli cells also have the function of doing support and providing the nourishment to all these developing spermatocytes. And super important, they regulate this process of spermatogenesis. So if there's a whole bunch of sperm hanging around that haven't been called to duty, they can slow this process down a little bit. All right. So let's look at spermatogenesis. Here is the outside part of the seminiferous tubule. We have a cell here that's first going to undergo mitosis, and you're going, why? Because I thought gametes underwent meiosis. Well, if a cell just underwent meiosis, then it would be gone, right? So we have to have a cell undergo mitosis first so that one of those two cells can undergo meiosis and the other cell sticks around so it can start later on. Otherwise, once the first cell was gone, you'd never have any more sperm made from that point on. All right, so first we have to have mitosis going on of the germ cell. Then we are going to have meiosis 1 take place, where that diploid cell will then become haploid. Then we're going to have those two cells at the end of meiosis 1, each divide, and we would end up with 4 cells at the end of meiosis 2. So let's see what that would look like here and what words we're going to use. So inside the seminiferous tubule, notice here in purple, we have our Sertoli cell completely surrounding. In green, all the cells, which are going to be our gamete cells. So we start here with our germ cell, which is called a spermatogonium. Okay, this is the cell that undergoes mitosis and forms 2 primary spermatocytes. Well, it's actually going to form 2 cells. One will be a primary spermatocyte because it's going into meiosis 1. So remember, meiosis 1 uses a primary spermatocyte. The other cell is going to hang out and it's going to stay as a spermatogonium so it can start the process again. All right. So now we are going to have meiosis 1, where our primary spermatocyte, which has moved away from the wall, is going to go from diploid to haploid and make 2 secondary spermatocytes. So in meiosis 2, we're going to use secondary spermatocytes, which are already haploid, to make 4 spermatids, each of which is going to have 23 chromosomes. Now, you know sperm don't look like this, right? So these are called spermatids. They're not sperm yet. Because as the last part of spermatogenesis, we have to do a little plastic surgery, a process called spermiogenesis, okay? Where we're going to take this round cell, we're going to make it look kind of like a badminton birdie with a tail, and then it's going to end up looking like a sperm. So let's look at this process of spermiogenesis, the final stage of spermatogenesis, when that spermatid develops into a mature sperm. Look at all the details of what has to happen to it. So inside that round or oval cell, what happens in the nucleus, the DNA all condenses and becomes nice and compact. The Golgi apparatus goes to work and starts making a truckload of digestive enzymes, just like you would be making a whole bunch of lysosomes. But this time the lysosome is going to go on the tip. So we take the prefix that means tip, which is acro, but instead of calling it a lysosome, we call it an acrosome. Okay, because what's going to happen is this acrosome is going to go over the top of the nucleus, kind of like pulling a hat over the top of your head. So it's called an acrosome cap or an acrosome vesicle, forming a cap over the top of the nucleus. At the same time, one of the centrosomes, you know, the thing that shoots out the spindles to pull the chromosomes apart. Well, in this time, it's going to take microtubules to form a flagellum. Yeah, the same thing is what makes the flagellum. Who knew? All right, and so it's going to start making flagellum, which is going to get longer and longer. And the other thing is going to happen, all the mitochondria in the cell are all going to start congregating in one area. And where they're going to congregate is going to be the area between the nucleus and the start of the flagellum. And after this, basically, there's going to be a little nip-tuck plastic surgery, and you're going to pull off all the extra cytoplasm and get rid of all the other unneeded organelles. So we're going to throw away the Golgi, we're going to throw away the endoplasmic reticulum. We're just going to get rid of everything else because this sperm means business. It's only going to take what it needs. Okay, and the last part is the head will taper more into a cone head. So yes, you can see some sperm that are really pointy, some that are not quite as pointy, and some that are more rounder in shape. So take a moment, stop the video here, and kind of go through that last step of sperm formation here. All right, so when you look at one sperm up close, let's look at the details, what we have going on. We can divide the sperm into three parts. We have the head at one end where we have the acrosome, which covers the nucleus. In the middle, we have what's called the midpiece, which is just chock full of mitochondria. And then we have the tail, which is just the flagellum covered with the plasma membrane. And notice there is no extra cytoplasm. So at the beginning, I talked about why the testes were outside, so to keep them cool. But can you have a higher temperature in the testes even outside? Yes, so let me show you some of the things that could cause that problem. For instance, what if the testes doesn't go all the way down in the scrotum? It's only part way down. Or what if you have an infection or inflammation? Both of those would raise the temperature. Sometimes there's extra blood vessels down there in the scrotum, or there's even a collection of extra fluid. Both of those would raise the temperature. So those are well-recognized medical things, but then there are non-medical things that would raise the temperature. Sitting with the laptop on your lap. Have you ever felt your laptop? It gets warm, okay? Or even if you're using a computer, if you are sitting down all day, you know, you need to cool those guys. Wearing tidy whities instead of boxers, all those things will raise the temperature of the testes. And the net result of that will be a decrease in the number of sperm, a decrease in the motility of sperm, change in how the sperm looks, some of those funny looking sperm, and an increase in the fragmentation. And what has happened is we have decreased the number of sperm in an ejaculate by 50% in in the last 25, 30 years here in the United States, and we have increased the number of the funny-looking sperm. So, lifestyle factors that affect the sperm. If a mom is smoking while she's carrying a baby boy, it affects his sperm. If you sit for more than 2 hours at a time, so even if you're standing up all day working, if you're a gamer, you're affecting your fertility. If you like to eat a high-fat diet, sitting, even if it's only having wings while watching football games, but that's potentially reversible if you change your diet. If you have shampoo and skincare, like you want to take care of yourself, so you start using a skincare regimen. If those things come in plastic bottles, those plastic bottles contain phthalates, and those phthalates interact with your hormone receptors. So even though you're making your normal hormones, they can't interact with their receptors because the phthalates found in those plastic bottles have blocked them. If you like to take relaxing hot baths or visit spas, that decreases your sperm count. And marijuana decreases the sperm count, but interestingly, alcohol and tobacco do not. So take this chance to review the process of spermatogenesis, and I want you to realize this entire process of spermatogenesis, the mitosis, the meiosis, and then the hanging out, learning how to swim, the backstroke, treading water, all that stuff that happens in the epididymis before being called to duty, that entire process takes 74 days. So if you're listening to this at the right time in the fall course, which is usually near the end, that means the sperm that are being made today will be ready to be called to duty for Valentine's week. Oh, so think about that if you're having sex around Valentine's Day. Or if you are taking this information in the spring course, the sperm are being called to duty about a week before the beginning of your fall semester. So, about the third week of August. So with that, I am done with this part. I only have one more video for the male reproductive system, and that is going to be on the male hormones. So I'll see you for that part. Once again, thank you for all your hard work.